Today we are going to be discussing chromosomal aberrations, which are chromosome mutations that change the structure of individual chromosomes. There are 4 types of aberrations which may occur, these being duplications, deletions, inversions, and translocations. These aberrations may arise when DNA breaks and cells attempt to reconnect broken fragments of DNA, or through errors made during crossing over. Crossing over occurs during the process of meiosis and among homologous chromosomes. In brief, meiosis is the pairing and separation of chromosomes and the division of sex cells to produce gametes. Synapsis refers to the close pairing of homologous chromosomes, while crossing over is the sharing of genetic information between 2 non-sister chromatids in a homologous pair. We are going to be examining the gametes produced by 2 individuals. The first individual has the chromosomes MLI centromere JKHG and MLKJ centromere IHG. On the screen, I have highlighted in blue the portion of the chromosomes which are altered, and in green the parts which are the same. It is observed that in the first chromosome, the order of the blue portion is I centromere JK, and in the second, it is KJ centromere I. The order of this portion is reversed, and because it contains the centromere, this aberration is a pericentric inversion. To predict the gametes which will be produced, an inversion loop is used, which depicts the reversed order of the blue portion of the chromosome and crossing over between the J and K region. From following along each chromosome, the gametes produced are predicted. The gametes which will be produced are MLI centromere JKHG, GHKJ centromere IHG, MLI centromere JKLM, and MLKJ centromere IHG. Only the 2 with check marks next to them will be viable, as the other 2 contain duplications and deletions of genes. In the second individual, there are no chromosomal aberrations present, so all the gametes produced are the same and viable. When comparing the fitness of these 2 individuals, individual 2 is more fit as all of the gametes produced are viable, while only 50% of the gametes produced by individual 1 are viable. This concludes my exam number 3 mini video. Thank you for watching.